Have you ever memorized the steps of skeletal muscle contraction, but during exam everything get mixed up? Calcium, troponin, tropomyosin, actin-myosin interaction, power stroke, ATP, and finally you don't know what comes first and what comes next? Don't worry. In this video, I'm going to make the entire mechanism into one simple sequence that you can understand, reproduce in the university answer, and remember during the exam. And stay till the end because I'm going to show you the master sequence of a skeletal muscle contraction from the arrival of nerve impulse to actual shortening of the sarcomere. Once you understand that sequence, the entire topic becomes much easier to recall. So let's start from very beginning. Today's topic is skeletal muscle contraction, specifically the sliding filament theory. Now, before we jump into calcium and ATP, let's understand one fundamental question. What actually happens when skeletal muscle contract? Does the actin filament become shorter? Does the myosin filament become shorter? No. This is the first concept you must remember. During contraction, the actin and myosin filament slide past each other, causing the sarcomere to become shorter. That's why we call it as a sliding filament theory. Think of it like a two combs sliding into each other. The individual filaments don't have to become shorter. The relative position changes. And this simple idea will explain everything we are going to discuss today. But there is one important question. How does a nerve signal ultimately causes actin and myosin to interact? That's where the few new slides come in. Let's first understand the anatomical setup, the triad. Before contraction begins, we need to know where the signal is going. Look at this diagram. The T-tubule or the transverse tubules are the invaginations of the sarcolemma. Their important function is to carry the electrical signal deep into the muscle fiber. Now beside the T-tubule, we are having terminal systems of the sarcoplasmic reticulum, that is L-tubule. The sarcoplasmic reticulum is basically the major calcium store of the muscle fiber. So we have one tubule plus two terminal systems, that is one triad. This is extremely important for exams and where this triad is located at the AI band junction. This diagram also highlights an important point. There are two triads per sarcomere. So remember, T-tubule carries the electrical signal, sarcoplasmic reticulum stores calcium, and the triad is T-tubule plus two terminal systems. Now here is the key question. If the nerve gives an electrical signal at the surface, how does the signal reach the calcium stores deep inside the muscle? That's called as excitation-contraction coupling. Let's say it step by step. Now let's move from anatomy to physiology. Skeletal muscle is an excitable tissue. That means it gets excited by electrical phenomenon in the form of action potential and respond by contracting in the form of mechanical phenomenon. The action potential travels along the sarcolemma and then enters the T-tubules. And remember what we just learned? The T-tubules carry electrical signal deep inside the muscle. The electrical signal eventually reaches the excitation-contraction coupling machinery. And this is the point where electrical signal is converted into a mechanical event. So remember this very simple phrase, electrical signal causing mechanical contraction. That is the essence of excitation-contraction coupling. Now, one very important exam point. The key idea is that skeletal muscle has a resting membrane potential of minus 90 millivolts, whereas nerve shows about minus 70 millivolts. The excitation threshold for skeletal muscle is 30 to 40 millivolts, whereas for nerve it is 15 millivolts. And also, the action potential for skeletal muscle is much longer than that of the nerve, and conduction velocity is lesser than that of the nerve. But don't get lost in the numbers. For this topic, the important concept is action potential reaches the tubules and excitation-contraction coupling begins. Now we need to understand what exactly happens at the T-tubules. Now we reach one of the most important mechanism in the entire topic, excitation-contraction coupling. This slide shows T-tubule, DHPR receptors and the ranodin receptors. Let's make this extremely simple. The action potential travels down the T-tubule. The voltage change activates the DHPR, that is dihydropyridine receptors. DHPR is mechanically linked to the ranodin receptors RYR present on the sarcoplasmic reticulum. And when DHPR changes its conformation, it physically pulls or activates the RYR receptors. The RYR then open, and what comes out? The calcium. So the entire mechanism can be remembered as action potential, DHPR, ranodin receptors open, and calcium release. Now pause here. If you are watching this for your university exam, mentally repeat the sequence once again. Action potential, DHPR, ranodin receptors, and calcium. That's your first major memory chain. But calcium itself doesn't directly pull actin. So what does calcium actually do? That's what we're going to see next. Now, the calcium enters the story. When the ryanodine receptors open, calcium is released from the sarcoplasmic reticulum into the cytoplasm, and the calcium concentration rises dramatically. That is about 200-fold increase in calcium concentration. But here is the most important question. What does calcium bind to? Calcium binds to troponin C, and this causes a conformational change in the troponin tropomyosin complex. And as a result, tropomyosin moves away from the active site on the actin. This is a very important concept. At rest, tropomyosin is blocking the active sites of actin, but after calcium binds to troponin C, tropomyosin moves and active sites become exposed. About 7 binding sites are uncovered per troponin calcium complex. So after calcium binds to troponin C, tropomyosin moves and active sites are exposed. So remember, calcium causes act with the troponin C, causes tropomyosin to move, and the actin sites are exposed. And notice something important. Calcium is basically the permission signal for concentration. It removes the block that was preventing actin-myosin interaction, and that forms the basis of excitation-contraction coupling. But now comes the real engine of contraction, the cross-bridge cycle. And this is where ATP becomes extremely important. Let's see how the power stroke actually happens. Now we come to the actual mechanical event, the cross-bridge cycle. It is also called as sliding filament or ratchet theory, given by F. and H. E. Huxley in 1954. It begins with step 1, that is attachment. The myosin head contains an ATPase, which later splits to ADP and phosphate group. In this energized state, myosin is ready to interact with the actin. Once the active site on the actin becomes exposed, the myosin head binds to the actin. That is, the head extends at 90 degrees and attaches to the actin. Now comes the power stroke. The myosin head changes its position and pulls the actin filament toward the center of the sarcomere. That is, when ADP and phosphate are released, myosin head flexes toward the arm to 45-degree angle. This is the actual mechanical movement that produces the contraction. That is, the flexion from 90 to 45 degrees generate mechanical force. Now look at the diagram. The myosin head moves from approximately 90 degrees towards the 45 degree. That movement generates the power stroke. So remember this. Myosin binds actin, power stroke, and actin moves toward the center. This is the part where the muscle actually generates force. But the story is not yet finished. After the power stroke, the myosin head is still attached to the actin. So how does it detach? That's the next step. After the power stroke, the myosin head remains attached to the actin. Now we need to release it. And this is where the ATP comes in again. A fresh ATP molecules bound to the myosin head. This causes myosin head to detach from the actin because myosin ATP complex has low affinity for actin, and this causes the myosin head to detach from the actin. This is an extremely high yield point. So remember, ATP binding leads to detachment, not ATP hydrolysis. ATP binding causes the myosin head to detach from the actin. After this ATP binding, next stage is reactivation. Here the ATP splits again, and when the ATP is hydrolyzed, it re-energizes the myosin head, preparing it for the next cycle. That is, the head extends back to 90 degrees for the next cycle. So the cycle becomes binding of myosin with actin, followed by power stroke, followed by ATP bind, lead to detachment, then ATP hydrolysis, and the myosin re-energized. And then the cycle can repeat as long as the calcium is present and the ATP is available. This is one of those places where the student often mix up ATP function. So keep this distinction very clear. ATP binding causes detachment, whereas ATP hydrolysis causes re-energization of the myosin head. Now, if thousands of these cross-bridge cycles happen together, what happens to the sarcomere? Let's see. Now let's look at the final structural result of all these cross-bridge cycles. That is, the sarcomere changes during contraction. But here is very common university exam questions. So let's see. Width of A band remains constant. Why? Because the length of the thick filament does not change. H zone disappears. I band width decreases and Z lines move closer together. As a result, the entire sarcomere shortens. This is extremely important for your exam diagram. So let's create one simple memory line. Remember AHIZ. A for A band, that is always same. H for H zone, that is happens to disappear. I is I band is reduced, and Z stands for Z line, that is zooms closer. and the entire sarcomere becomes shorter. Pause the video for 5 seconds and see if you can recall all 4 changes without looking: A band, H zone, I band, and Z lines. If you got them, excellent. Now we know how contraction occurs, but eventually the muscle has to relax, and the relaxation brings us back to calcium and ATP. Now let's understand relaxation. When the stimulation stops, calcium must be removed from the cytoplasm. That lead to relaxation. This calcium is actively transported back into the terminal systems. So the removal of this calcium from troponin C, it restores blocking action of tropomyosin. That is, the active sites on the actin becomes covered again. The cross bridge formation stops and the muscle relaxes. So the relaxation can be remembered as calcium removal, then troponin C loses this calcium, then tropomyosin blocks actin, cross bridge stops, and the relaxation occurs. But this slide has another very important exam concept: the 3 important functions of ATP. First one, ATP hydrolyzes the cross bridge and gives the energy. The binding of ATP causes dissociation, that is, the detachment. And ATP also gives energy for calcium ATPase pump, that is responsible for relaxation. So ATP is not a simple energy, it has multiple important roles. And if this ATP is absent, something interesting happens. The cross bridges cannot detach. That lead to one of the important or most fascinating applied aspect of this topic, that is the rigor mortis. Now let's connect physiology to the real life application. There is shortening and rigidity of all the body muscles soon after death. The cause is total loss of ATP, because after death the ATP production stops, and without fresh ATP the myosin heads cannot detach from actin and the cross bridges become permanently fixed. This is called as rigor mortis. So look at this sequence. There is a death, then ATP production stops, myosin cannot detach from actin, and the muscles become rigid. And that's why the key statement is no ATP, no detachment lead to rigidity. This is also a simple or beautiful example why understanding ATP is more important than simply memorizing its role. But remember, this rigidity disappears after some hours due to muscle protein destruction by cellular lysosomal enzymes. Now let's connect everything. We have traveled through nerve excitation, then T-tubule, dihydropyridine receptors, ryanodine receptors, calcium, troponin, tropomyosin, and actin-myosin interaction, then power stroke, the ATP, then sarcomere shortening, and finally the relaxation. But if you have to reproduce this entire mechanism in the exam, you need one single master sequence. That's exactly what we're going to build in the next slide. Now, this is the slide I want you to remember before entering your physiology exam. This is the master sequence of skeletal muscle contraction. Let's go through it once from beginning to end. Step 1. Nerve excitation. The nerve is stimulated and action potential is generated in the motor neuron. Step 2 is conduction. The impulse travels down the motor neuron and reaches the neuromuscular junction. Step 3. Neuromuscular transmission. Acetylcholine is released at the neuromuscular junction and it binds to a receptor on the muscle membrane and it produces an end plate potential. Step 4. Muscle excitation. The action potential spreads along the sarcolemma and enters the T-tubule. Step 5. Excitation-Contraction Coupling. The action potential activates dihydropyridine receptors. DHPR activates the ryanodine receptors and the calcium is released from the terminal systems of sarcoplasmic reticulum. Step 6. Calcium action. Calcium binds to troponin C. Tropomyosin moves away from the active sites of actin. Step 7 is muscle contraction. Myosin bind to actin, cross bridge formation occurs, power stroke occurs, and the actin slides towards the center of the sarcomere, and the sarcomere shortens. That's the entire story. Now, I want you to look at the sequence as one chain. Nerve excitation, acetylcholine release, muscle action potential, T-tubule, DHPR, ryanodine receptors, calcium release, troponin C, tropomyosin moves, actin-myosin interaction, power stroke, sliding of actin, and the sarcomere shortening. If you can reproduce this chain, you can explain the entire mechanism logically instead of trying to memorize isolated sentences. If this explanation helped you understand the skeletal muscle contraction instead of simply memorizing it, make sure you save this video for your exam. And if you are preparing for your MBBS Physiology University exam, subscribe to PhysioWithNeo MBBS Exam Simplified because here we focus on understanding concept and converting it into an exam-ready answer. If you want to revise the same topic in a 5 minutes revision, see the next video of skeletal muscle contraction rapid revision in 5 minutes. And if you want to memorize the sliding filament theory, ratchet theory in short time, see this memory trick video. Till then, keep studying smart and keep your physiology simple.